I would like to invite Taiji Rama for presenting the topic, or presenting the presentation on potential role of biosurfactant as nutraceutical in ROS-reduced skin damage. Yes, Mr. Taiji, can you please give your brief introduction?
Ma'am, am I visible?
Yes, please.
Okay, thank you. So, good afternoon to one and all present here. First of all, I'd like to express my gratitude to be part of this virtual symposium. Today, I'm going to discuss my title, The Potential Role of Biosurfactant as Nutricosmeceutical in ROS-induced Skin Damage. So, these are the contents. So coming to the introduction part, skin damage, as we all know, is defined as uneven skin tones such as pigmentation, age spots, redness, acne. The common causes of skin damage includes sunburn, cancer, acne, bleaching, physical accidents, or cuts. Now, skin aging can be characterized by loss of functionality and resistance of adaptability to stress and age-associated diseases. Now, the another important factor which can cause skin damage is microorganisms, which are implicated in a number of infectious and non-infectious skin conditions, which affect the various layers of the skin. Now, skin aging have become a global phenomenon in the 20th century. There are two different types of biological theory of skin aging, stochastic theory and non-stochastic theory. Stochastic theory suggests that skin damage is caused by free radicals, random errors, and non-stochastic theory suggests that skin aging is controlled by genetic programming. Now, the various intrinsic and extrinsic factors of skin aging are: the examples of intrinsic factors includes ethnicity, hormonal changes in cutaneous tissue, and examples of extrinsic factors are lifestyle influence, effects of smoking, and overexposure to UV radiation. In the current scenario, overexposure of UV radiation have been identified to be an important role in the skin aging process. Now, how this UV radiation is going to cause a skin aging? UV radiation can induce reactive oxygen species, which is a highly reactive molecule containing oxygen derived in living organisms as byproduct of aerobic activities. They are often known as dual-edged swords because ROS can occur normally in the living system as a part of the signaling pathway. On the other hand, overexpression of reactive oxygen species can cause harmful effects on the body by damaging various macromolecules. The examples include superoxide, hydrogen peroxide, hydroxyl radical, and etc. Here, the figures show the different external factors responsible for ROS production. So, the reactive oxygen species can attack the normal cells, which lead to various macromolecular damage under a condition known as oxidative stress. Here, the different signs of photoaging have been shown. So, this is one of the molecular mechanisms of skin aging by reactive oxygen species, which have been reported by many literature. So, here, the UV radiation by sunlight can induce reactive oxygen species formation, which can go and bind with the cytokine receptor on the skin cell membrane. This can then activate two signaling pathways. One is the MAP kinase pathway and another is the nuclear factor kappa B. So these two different signaling pathway can go and bind with the MMP promoter, which leads to the activation and production of different MMP protein, which is responsible for collagen degradation and photoaging process. Now, similarly, here the mechanism of skin damage caused by disease-causing bacteria have been shown. In the normal skin, the different layers of the skin is protected by microbiological barrier hydrolipidic film. However, under stress conditions or when the normal skin is attacked by allergens or pathogenic bacteria, the microbiological barrier hydrolipidic film is damaged, which causes the skin to lose its moisture over time, leading to the skin damage. Now, in order to counteract the skin damage caused by reactive oxygen species and certain microbes, the idea of developing nutricosmeceutics have been adopted. Nutricosmeceutics, it's a latest term in beauty industry formed by amalgamation of the term cosmeceutical and nutraceutical. It is basically a bioactive compound uh, which is intended for health, which is intended for our health benefits. So the development of nutricosmeceutics basically involve a delivery vehicle for bioactive compound. In our case, a biosurfactant is the bioactive compound which have been extracted from gram-positive Cochuria marina DAC2, which was previously isolated from the soil of NIT Durgapur. So here the important role of antioxidant against reactive oxidant species have been shown. Basically, there are two different types of antioxidant, endogenous antioxidant and exogenous antioxidant. During the aging process, the endogenous antioxidant present in our body declines. Hence, the importance of exogenous antioxidants comes into the play. Hence, nutritional supplements containing exogenous antioxidants becomes important to protect ourselves against ROS-induced skin damage. Similarly, the mechanisms of antimicrobial compound on human skin have been shown here. Biosurfactants can be used as an antimicrobial agent to prevent microbial growth on the skin surface. They can form pore-bearing channels by self-associating their molecules to trap the microbe in between the aggregates, which prevents their growth. Now, these are the different applications of biosurfactant.
You need to stick to the time. You have another 5 minutes to go.
Oh, okay, ma'am. Okay, yes, ma'am. So these are the approach to overcome the challenges of biosurfactant. So in our case, we have chosen hydrogel as delivery vehicle due to its several advantages. These are some of the important properties of hydrogel, which makes it one of the good delivery vehicle for topical system. So these are the materials and methods. Coming to the results and discussion part, first of all, here, the screening results for biosurfactant production by different methods have been shown. From oil spreading assay and drop collapse assay, the positive results of biosurfactant presence have been detected. Here, the addition of biosurfactant can reduce the oil surface tension in oil spreading assay as well as drop collapse assay. Next, C-TAP assay was performed, which showed the presence of biosurfactant by forming a dark blue color surrounding the wells. Then we extracted a biosurfactant from the cell-free supernatant, and the absorbance spectra of extracted biosurfactant showed maximum absorbance at 265 nanometer wavelength. Then characterization of extracted biosurfactant was done by TLC analysis, which showed the presence of sugars, amino acids, and fats. FTR analysis was also performed, which showed the presence of different functional groups present on the biosurfactant. HNMR and CNMR analysis have also been performed, which show the presence of aliphatic chains, presence of aromatic compounds. CNMR indicated the presence of different alkane groups and tertiary carbon atoms. Thus, the characterization studies showed the complex glycolipid nature of the biosurfactant. Next, antioxidant activity of biosurfactant was performed by DPPH assay and Abitase assay, which showed its radical scavenging activity. Then antimicrobial activity of biosurfactant was performed against 3 different bacteria. Here, the biosurfactant showed its antibacterial activity at increasing concentration of 32 mg per ml. Then we have performed preliminary study for hydrogel formulation by using different ratio of sodium alginate and gelatin, and from the tube inversion method, a 2:5 ratio of alginate and gelatin showed good and stable gelling property. Then we performed porosity test, and from the porosity study, these are the results that we have obtained. So based on the these two results, 2:5 ratio of sodium alginate and gelatin have been selected to further encapsulate different concentration of biosurfactant. Then here swelling activity of hydrogel have been performed, and it was observed that hydrogel encapsulated with 16 mg per ml biosurfactant showed a good swelling percentage followed by 32 mg per ml of biosurfactant. Then a further study was performed to understand the bioactivity of hydrogel encapsulated biosurfactant, and it has been observed that hydrogel encapsulated with biosurfactant has good scavenging activity towards DPPH and Abitase radical. Then we also perform antibacterial activity of encapsulated hydrogel with biosurfactant, and it has been observed that biosurfactant encapsulated with 16 and 32 mg per ml of biosurfactant has good antibacterial activity against the 3 different bacteria. Now, based on the work, I would like to conclude that the present work confirmed the biosurfactant production by Cochrane Marina DEC2 presence of different compounds was confirmed by the TLC analysis and different characterization methods. Radical scavenging activity of biosurfactant showed its potential application as antioxidant agent. Good swelling and porosity indicate the water holding capacity of hydrogel, which may allow better release of metabolites. Antioxidant and antimicrobial activity of hydrogel-loaded biosurfactant indicated its potential application to target skin damage. However, further research should focus on optimization, characterization, and stability of developed hydrogels. These are my publications. Lastly, I would like to thank my supervisor, Dr. Devjani Datta, for her guidance, immense support, and motivation in carrying out this work. Thank you so much.
Thank you, Ms. Taiji. It was a very nice talk.